Okay, welcome back. Now we're going to talk about eukaryotic cells. Okay, we talked about prokaryotic cells. We talked about the history of cells in general. Now, eukaryotic cells, um, they're much more complex than prokaryotic cells. They're typically larger than prokaryotic cells, but the complexity really comes in what's held within inside that cell membrane, that plasma membrane that we've talked about thus far. Okay, so larger and more complex, but the complexity really comes in organelles. Okay, so organelles are compartments, membrane-bound compartments that have some kind of function. In some cases, maybe that compartment deals with just making proteins. In other cases, that compartment might deal with making fats. In other cases, maybe it deals with breaking energy down. In other cases, maybe it deals with breaking foreign material down. Excuse me. So, in all cases, these organelles are these compartments that have some kind of function for the cell. Okay? The other component is a much larger, elaborate cytoskeleton. So inside eukaryotic cells, there's a lot more protein fibers that make up kind of a scaffolding inside the cell. So it keeps the cell a uniform shape typically, and there's a little flexibility there still. You have to have the flexibility. But the key component with the cytoskeleton is it provides little regions where those organelles can kind of bind to. So they're not just all floating to one side of the cell and or bunching together. That they can move within the cell through the cytoskeleton structure. And on top of that, it also allows for proteins to be packaged and moved along these protein fibers or these scaffolding proteins. Okay, so most of the time when we're looking at things under a microscope, there are some organelles that you can really see under something like a compound light microscope. And so the next pictures that you'll see, and we're going to go through all the organelles that are really truly important for, say, allied health. To know what would happen if your mitochondria is malfunctioning. What does that do to the individual? What happens if you accidentally get exposed to cyanide? What happens, all these scenarios are at the cellular level. And so knowing what's going on in the cell will give you an idea of what might happen, what kind of experience, and what kind of haywire is going on. Okay. The first thing we're going to talk about is the nucleus, and by far this is probably the most important. Okay, so again, nucleus, it the term itself means true nut. Okay, so that's where, remember, eukaryotic, true nut or true center. Nucleus means it has a true nucleus, a true nut, a true center. Okay, and so that nucleus is a membrane-bound compartment that houses DNA and RNA, for that matter, and ribosomes. Okay, they have an endomembrane. Okay, so it's this area, this nucleus, is covered by a membrane specific to itself. Okay? And that membrane is called the nuclear envelope. And the reason why it's specific to itself is because often there's proteins that are only associated with that endomembrane. And so if you go back to that membrane that I showed you, that fluid mosaic, and you saw all those phospholipids sitting in there, and then you have these proteins that are going through those phospholipids. Same thing in the endomembrane. except for those proteins, may not occur anywhere else except for that membrane. Okay. Now, eukaryotic cells, being that they're larger, more diverse, more complex, also gives us rise that we can have different types of eukaryotic cells. And so this is a good thing because cells have to have different functions. And so, for example, plants, fungi, some protista, and of course, bacteria, they have cell walls. Now, the plants and fungi and protista have a cell wall that's made different. It's different than the bacteria, but they have a cell wall that's different from each other. Right? So, for example, plant cell walls are made out of cellulose, which is a carbohydrate. Okay? Fungi cell walls are made out of chitin, okay, which is also a carbohydrate, but they're not the same. Okay? And protista can have either. And so, but that cell wall serves the same purpose as the cell wall in bacteria and prokaryotes. It's an extra piece of protective material. The difference here, though, is the plant and fungi and protista cell walls make them rigid. Okay, so they're more rigid. This is what provides plant cells with that rigidity and allows them to get 300 feet in the air. Okay, is they lose their flexibility. Okay? They still have a cell membrane underneath it, which is flexible, but the wall itself is not really flexible. Okay? Plants, protista, organisms that can do photosynthesis, they have organelles that are called chloroplasts. That's a site of photosynthesis. Okay? Plants can also have what we call a central vacuole, okay, which is this large sac-like membrane organelle, which can house a bunch of nutrients, so calcium and things like that can be stored in there, potassium, nitrogen, okay? But it also allows for them to maintain a lot of water for turgor pressure. So the cells can hold a bunch of water in their central vacuole. Animal cells, we have a specific organelle called the centriole, and the centriole is this organelle that generates, or it's a site where microtubules generate from it, important for mitosis and meiosis, which is how cells replicate themselves. Okay. So here's an animal cell, lots of different organelles. See, if you look at this, it's very different than a prokaryotic cell. So this is a example of a eukaryotic cell, an animal cell. Still have that membrane, still very similar membranes in the sense that it's phospholipid bilayer, there's proteins embedded in it, okay? But now you have all these other structures in here which have their own membranes, endomembranes. Okay? And those are all called organelles. So whether we're talking about mitochondria, Golgi apparatus, the lysosomes, okay, all those things will have their own membrane and have their own function for that system. Okay? All right, so let's dive into the nucleus. Okay? So the nucleus, highly organized. The nucleus is surrounded by a double membrane, okay? So a lipid bilayer called the nuclear envelope. Okay? There's also another region inside the nucleus called the nucleolus. Okay? The nucleolus is where ribosome RNA is made. So it's basically the site where ribosomes can be constructed. Okay, so you can build new ribosomes in the nucleolus or associated with the nucleolus. Okay, so here's just an example of it. Okay, so you can see this outer membrane. Okay, that's the nuclear envelope. So this would all be within inside the cell. Okay, so you get inside the cell, you find the nucleus. It has a nuclear envelope. Okay, it has a nucleolus, this region where ribosomes are regenerated, and then it has, because this is where DNA is housed, it will have chromosomes, which is how DNA is packaged. DNA is packaged in chromosomes, which are just long strands of DNA wrapped around proteins and kind of form these structures that are more condensed. Okay. All right. The rough endoplasmic reticulum. Okay. So the rough ER, it gets its name rough or R. Okay. So sometimes people will call it the RER. Some people will call it the rough ER. Okay. Some people call it the rough endoplasmic reticulum. Okay. The rough part is ribosomes. Okay, so there's ribosomes on the outside of this membrane. So associated with the RER are ribosomes. Okay, the interior inside that structure is called the lumen. Now, what's its function? Well, it's a site, one of the very important sites of protein synthesis. Okay, so remember, it's associated with ribosomes, and we already said ribosomes are where proteins are being made. So you have all these proteins attached to the rough endoplasmic reticulum, and proteins are being made. Those proteins are being made, and they get put in vesicles and little parts of the rough ER, and then that material, that sac with proteins in it, can be pushed away to a different feature. Okay, so here you can kind of see it. Okay, so just dotted with lots and lots of ribosomes, and there are these kind of sacs, membranes with, filled with lumen. Okay, and then you can see away from it here, here, here you can see these vesicles, and here in this picture you can see these vesicles that have broken off, chunks of the endoplasmic reticulum that are broken off, and inside that would be proteins. And they're going to travel to typically the Golgi apparatus, which we'll talk about here in a second. All right, now let's look at another one that's not the rough ER, but the smooth endoplasmic reticulum, or the smooth ER. Okay, there's no ribosomes. That's the difference. Okay, but the other thing is, since there's no ribosomes, they're not making proteins here, but instead of making proteins, it's making fats or assembling new phospholipids or breaking down fatty acids. It also breaks down poisonous lipids. Okay, it breaks down lots of poisonous substances, and it can store things that are useful for building other material, whether that be building more fatty acids, phospholipids, so it can store phosphates. But one of the big ones that it stores is calcium. Okay, and here you can kind of see it. You can still see vesicles because, again, it's generating things like phospholipids and whatnot, and they'll be embedded in a vesicle, and those vesicles can leave, but there's no ribosomes associated with the smooth ER. So we had that nucleus, we have that rough ER, and then also associated with that nucleus, you got the smooth ER. Okay, so now those vesicles, they gotta go somewhere, and where they're going is the Golgi apparatus, most likely. Sometimes they can go straight to the membrane or straight to where it's needed, but often it's going to the Golgi apparatus, because the Golgi apparatus is like the UPS store. You bring your package in, they repackage it, make sure that it's going where it needs to go, and then send it out. Okay, and so it, again, is just a bunch of membranes stacked together, okay, and we call that cisternae. But the purpose of it is really to ship things to different parts of the cell, ship proteins most of the time to different parts of the cell. Okay? And vesicles will carry out that deal. So you have the rough ER, sometimes the smooth ER. Okay, they're going to send these vesicles into the Golgi apparatus. And so things enter what we call the cis side. Okay, so the vesicles will enter the cis side, they get repackaged, and one of the key components here, this repackaging, is stamping them with carbohydrate. Stamp it with the sugar, okay, to show that it was self-made, especially on those proteins that might be going to the outer membrane. Okay, a lot of those proteins, they got to be self-made, self-identifying markers, so your immune system doesn't attack it. Okay, and so it would come through, get rearranged. Sometimes the proteins need to be folded a different way, so folding might happen here. They get stamped with the sugar often, and then boom, they get back in vesicles on the trans side and pushed out to wherever it needs. That pushing, what's not really shown here, is you have protein fibers, microscopic protein fibers, that these vesicles can attach to and ride through the cell on these protein fibers. So long protein fibers that might go all the way to the cell membrane, and if that vesicle needs to put a protein on the cell membrane, it would just attach to that fiber and then head out all the way from the Golgi apparatus to the outside membrane. Okay, so ribosomes, again, we talked about this before, they're not organelles. Okay? But that doesn't mean they're not important. They're super important. Every organism, no matter what the organism is on this planet, has ribosomes. Okay, so that's one of the key features. Every cell that's ever been discovered has ribosomes. Okay? Because ribosomes are the site where proteins are being made. Okay? They have a large subunit and a small subunit, and RNA will go in between them, and that RNA code tells the ribosome or a different set of RNA exactly what amino acid, so protein's just a bunch of amino acids bound together, okay? But it tells exactly what amino acid to lay down in a sequence, okay? And so super, super important for protein synthesis, okay? Really can't emphasize it enough that they're not organelles. They don't have their own membranes, and they're two subunits. So right now it shows these two subunits bound together, but they occur, unless there's messenger RNA, they occur separately. Okay, you have a large subunit and a small subunit that would just be in the cytoplasm or just be on the rough ER, something like that. Okay. All right, lysosomes. Lysosomes are like the recycle center for the cell. Right? And so it's kind of like an internal immune system. Lysosomes, their job is to digest things. Okay, so they can digest foreign bodies. So if something sneaks into the cell that shouldn't be there, the lysosome can release digestive enzymes. Okay, digestive enzymes just a fancy word for a protein that can break up other things. And so some of these proteins can break up fats, some can break up carbohydrates, some can break up other proteins. Okay, so most of the time these lysosomes are going to have lots of different types of enzymes, digestive proteins, and they can be released on that foreign body. So something comes in, maybe a parasite like malaria gets into your red blood cells, and now these lysosomes can attack it and deposit digestive enzymes trying to destroy it. Okay. Other times though, it's not a foreign body. Sometimes it's your own material. Let's say you have a mitochondria or something that's old and it's not really functioning very well and needs to be dissolved, needs to be broken up. The lysosome will deposit digestive enzymes, and destroy it and break it up into pieces and then reuse the pieces. Okay, so it's kind of like a recycle center. Okay, and the final one I want to talk about is the mitochondria. Okay, and most people know this as the powerhouse of the cell. And the reason why it gets that name is because this is the site of ATP production. ATP, or adenosine triphosphate, is the currency, is the energy for the cell. It's your energy. So when you consume protein, carbohydrate, fats, all of it is converted to ATP because your cells have to use ATP to drive work. Okay, whether that be muscle cells, neurons, digestive cells, blood cells, whether that be red, white blood cell, it doesn't really matter. Their currency is ATP. That's what they have to use for energy, and the mitochondria is how that works. That's the site at where ATP is really made. Okay, so the mitochondria, they have these cristae, these like folds. Again, we talked about surface area to volume. Okay, even though it's kind of a circular structure with surface area limitations on the outside, but look at the surface area on the inside. So to increase the surface area, they have all these membrane folds, right? And all along there, you would have proteins that are embedded that allow for this structure, this organelle, to produce ATP. Okay. We can talk later about some things like that are unique to things like mitochondria and chloroplasts and some other things. They have their own ribosomes, so they actually have their own genetic material. So mitochondria has a circular chromosome that's associated just with the mitochondrion. And we can talk about what we call endosymbiosis, or how we think this might have came about at a later date. Okay, but for now, just knowing those organelles are fairly important.